Assalamu alaikum. So in this lecture, inshallah, we'll be talking about metabolism of heme. So again, resources, this lecture, and Lipin Class Biochemistry, the 7th edition, chapter 21, is really excellent, and you can refer to it. So let's go over the structure of heme first. We covered it in the first lecture, but it's not bad if we do it again. So basically, heme is a protoporphyrin molecule with a conjugated iron as a prosthetic group. And iron has to be in the ferrous state in order for heme to be active. Now the porphyrin again is planar. It consists of 4 pyrrole rings and they're designated as A, B, C, and D. Now each ring contains 2 substituents. You can have a vinyl group, a methyl group, and they can alternate. So you can have methyl, vinyl, and so on. And heme also has 2 propionate groups as well. Now, the molecule is quite hydrophobic, and iron has 6 coordinates of binding as discussed earlier. So heme and its intermediates are known as porphyrins, and these porphyrins are all made of these 4 pyrrole rings and different substituents. The pyrrole rings are connected to each other via methynyl bridges. Okay. Now, so you can have these substituents right here alternating, so you can have them as AP, 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 and so on, or they can differ as well. So you can have AP, AP, and then you have them as PA. So you can have them reversed on a single ring. Now, this gives the molecule a different name. So here you have the name of a molecule uroporphyrin 1, okay, having these acetate and propionate groups. And when you have the reverse order right here on a single ring, it's given the same name but a different number. These porphyrins are made of precursors, and the precursors are known as porphyrinogens. The difference between the 2 types of molecules is that porphyrinogens are reduced. That is, all of the nitrogens in the rings are all reduced. On the other hand, if you look at porphyrins, you have 2 of them being oxidized and 2 that are reduced. Okay, so these porphyrinogens are precursors of porphyrins, and porphyrinogens are colorless as well. Okay, but if they get oxidized, they would develop a color, and the color is purple, and that's why they're known as porphyrins. Now, and these porphyrinogens are intermediate molecules in the synthesis of heme.
Now, so let's start talking about synthesis of heme. Now, there are 2 major tissues that are responsible for the majority of heme synthesis, and these are liver and erythrocyte producing cells. Now, in liver, heme is used for production of hemoproteins such as cytochrome P450 enzymes. Now, the rate of heme synthesis is highly variable depending on the presence of certain drugs or as needed. On the other hand, in erythrocyte producing cells, the production of heme is constant. Okay? Now, inside cells, synthesis starts in the mitochondria, then it continues in the cytosol, and it ends in the mitochondria again. So what you have in here is a succinyl coenzyme A, which is a product, an intermediate of the citric acid cycle. It conjugates with glycine into a product known as aminolevulinic acid. This ALA then is transported out of the mitochondria it gets converted into several intermediates, and then one of them gets back into the mitochondria, and eventually it gets converted to heme.
So let's talk about certain reactions in a little more details. So the first reaction is probably the most important one, which is basically a reaction that is catalyzed by an enzyme known as aminolevulinic acid synthase. Okay, and there are two types, ALAS1, which is synthesized, expressed in all tissues, including liver, and you have ALAS2, that is erythroid specific. Now, ALA synthase catalyzes the conjugation or the condensation of glycine and succinyl coenzyme A into a molecule known as aminolevulinic acid. Now, this reaction is very important because it is the rate-limiting reaction or rate-limiting step and the committed step as well. So rate-limiting reactions are reactions that are slow because they are highly regulated. So the enzymes that catalyze these reactions are highly regulated, and this is the case for this particular reaction. And this reaction is also the committed step because once you have the formation of aminolevulinic acid, it has to go forward till the end, that is the formation of heme. Now, this reaction is also catalyzed, or it requires vitamin B6 or pyridoxal phosphate. And the reaction occurs in the mitochondria. Okay, and once it happens, ALA leaves the mitochondria to the cytosol, where you have the second stage of synthesis of heme. Now let's talk about the ALA synthase isoenzymes a bit more. So ALAS2, that is the erythroid specific enzyme, is regulated by the level of iron. Okay, it's also regulated by hypoxia. So if you have a patient with defective ALAS2, so there's loss of function, that results in what is known as, it results in anemia, of course, because you don't have the production of hemoglobin, and it's specifically known as X-linked sideroblastic anemia. The reason is that histologically you have the production of a ring staining right here in the erythroid cells, and it's mitochondrial as well, and it's known as mitochondrial non-ferritin iron ring sideroblast. Okay, so you can see them here and here as well. Now, ALAS2, particularly we're talking about the liver enzyme right here, is regulated by Heme. Okay, what is Heme? Well, it's the molecule, this molecule right here, that has iron in the ferric state, and it's linked to chloride. Now, Heme, what it does is four things. It regulates ALA synthesis by four mechanisms. It reduces transcription of the enzyme, it reduces the stability of the messenger RNA, it inhibits mitochondrial import of ALAS1, and it induces protein degradation as well. Now, ALAS1 in the liver is also regulated by drugs, certain drugs, because what these drugs do is that they require cytochrome P450 enzymes for detoxification or for modification. And that results in increasing the synthesis of these cytochrome P450 enzymes. Since there is now high demand for heme, that would reduce the level of heme in hepatocytes, and that would induce ALAS1 synthesis at the transcriptional level. Now, the following reaction is basically the condensation of two ALA molecules into an intermediate known as porphobilinogen. Now, this reaction is catalyzed by an enzyme known as ALA dehydratase, and this enzyme requires zinc, or it has zinc in its active site. Now, as a result, the presence of lead would result in inhibition of this enzyme, because what it will do is that it would replace zinc in the active site. So that results in accumulation of ALA in the body, and it would result in anemia, which is, of course, caused or associated with lead poisoning. Then what happens is that you have 4 molecules of porphobilinogens condensing into a linear molecule known as hydroxymethylbilane. Hydroxymethylbilane is a linear molecule, and it gets cyclized into uroporphyrinogen 3. Now this reaction is important because of what we will talk about later on. Now uroporphyrinogen 3 is converted to coproporphyrinogen 3, and coproporphyrinogen 3 moves into the mitochondria. Now, so as I said before, the reaction starts in the mitochondria. The first reaction takes place in the mitochondria, this reaction right here. Okay, and then ALA moves out. You have these reactions taking place in the cytoplasm, okay, until you get the formation of coproporphyrinogen 3, which moves back into the mitochondria. Now, then it gets converted to protoporphyrinogen 9, and it gets oxidized. So it's almost like this is a precursor, right? So we talked about this before, porphyrinogens and porphyrins. So it gets oxidized and it gets converted to protoporphyrin 9, which can have iron attached or conjugated to it, forming heme. Now this attachment or conjugation of iron into protoporphyrin can happen spontaneously, but it gets catalyzed by an enzyme, ferrochelatase. Now ferrochelatase is also sensitive to lead. It can be inhibited by lead. So lead poisoning, not only that it leads in the inhibition of the formation of the porphobilinogen and accumulation of ALA, but it also results in the inhibition of the very last reaction. Now someone might say, well, wait a second, why is that important? Because if you have this reaction inhibited, it would lead to there would be no intermediates, but this is not really the case, okay? Because you can still have some enzymatic activity right here. Okay, so you can have some intermediates, but of course at very low level. So lead can really terminate the synthesis of heme by inhibiting the enzyme ferrochelatase. So you have a sequence of reactions, and they are enzymatically catalyzed. So you can have mutations in any one of the genes that catalyze any of these reactions, and that would lead to accumulation of intermediates, and eventually, of course, you would get inhibition or low level of heme being synthesized. So that would result in a group of conditions known as porphyrias, and porphyria really means purple, and the reason is that these intermediates, some of these intermediates can be turned into the purple color, and you will have these intermediates, by the way, accumulating in serum or urine. Now, the disorders can be classified according to the affected tissue. Okay, so someone might say, well, and mainly we're talking about liver, we're talking about skin, and we're talking about erythroid tissue as well. Okay, so someone might say, well, wait a second, but all of these reactions take place they're needed in liver and they're needed in erythroid tissue, and the answer is yes, but there are certain variations in terms of how these enzymes are regulated in different tissues, how much they are expressed, how much they're needed, and the conditions. That is there a certain drug, for example, that would affect, of course, the enzymes in the liver? Okay, so and so on. So so they can be classified as erythroid-specific diseases or conditions, or hepatic. And hepatic can be acute or chronic as well, okay, depending on different circumstances the patient lives in. Again, ingesting certain types of food, taking certain drugs, ethanol, alcohol, for example, and so on. The disorders can also be classified according to manifestations. Okay, some conditions are photosensitive, others are not. Now that depends on which enzyme is inhibited, which one of them is blocked, which one of them is mutated. So this reaction is quite important. This is the reaction that converts hydroxymethylbutane, the linear molecule, into a cyclic molecule. Okay, that is the uroporphyrinogen 3. Now, if a mutation takes place in the enzyme that catalyzes this reaction and the previous ones, then we're talking about a not photosensitive condition. Okay, but because uroporphyrinogen and the intermediates that take place that are formed afterwards, they can absorb light and they can turn into the purple color, the patients would be more photosensitive. The reason is that you have accumulation of these porphyrins into the skin, and upon exposure to sunlight, you have the production of superoxide radicals that cause skin damage, cell death, and that would result in skin itching and burns, and it would look something like this. You would have a lot of blistering in the hands and areas that are exposed to light. Now, conditions where you have that are not photosensitive, the patients would have abdominal and neuropsychiatric symptoms. So this is taken from Lippincott, and we can talk about all of these conditions, but it doesn't make sense, of course. Now, these conditions, as I said, can be erythroid specific, as symbolized in the figure, or they can be hepatic as well. Okay, so I'll just focus on 3 of them. Okay, so we'll focus on lead poisoning. Okay, so we talked about it, and there are 2 enzymes that are that are affected, and it's the reaction, the ferrochelatase and the ALA dehydratase enzyme. So if this enzyme is mutated, or so is blocked or inhibited by lead, you can have accumulation of the ALA. If this enzyme is blocked by lead, you will have accumulation of all of the other intermediates. But the affected tissue mainly is the erythroid tissue. Now, you can have mutations in the ferrochelatase enzyme, and that would cause a condition known as erythropoietic protoporphyria. This is, it can be autosomal dominant or recessive, and you will have accumulation of protoporphyrin 9 in plasma and urine as well. And patients are, of course, photosensitive because you have accumulation of these intermediates. And the third one that we want to focus on is Porphyria cutanea tarda, and the reason is that it is the most common porphyria. Okay, so this is the one that you might be exposed to in the future. This is an autosomal dominant disease. It results in deficiency in the enzyme uroporphyrinogen decarboxylase that results in the formation of coproporphyrinogen 3 that goes into the mitochondria. Patients are photosensitive because this is a cyclic molecule. Okay, now yeah, you can have something known as hepatic porphyria, and that results, of course, in the low production of heme. But that results, this is something important, because heme is a regulator of ALAS1, the enzyme, the ALA synthase 1, low level of heme would result in increasing the expression of ALA synthase 1, and that results in increasing the level of all of these intermediates, and that results in increasing toxicity. And of course, you can imagine what would happen if certain drugs are ingested by these patients. So what's the best treatment? Well, the best treatment is giving patients Heme, since it inhibits the synthesis and activity of the ALA synthase. Another option is to give patients glucose. The reason is that insulin or glucose increases the level of insulin. Insulin inhibits the production of the transcription factor PGC1 alpha, which is needed for the synthesis or expression of the ALA synthase 1. So you can imagine what fasting would do. Fasting would increase the level of glucagon. Glucagon would induce the expression of PGC1 alpha, inducing the expression of ALA synthase, and of course that would result in increasing the levels of all of the intermediates.
So let's go into the second half of this lecture and talk about catabolism of heme.
So we said before that red blood cells are the largest storage place of heme. It contains, as we said before in the lecture Metabolism of Iron, that you have about 2,600 or 2,500 grams of iron in red blood cells. So that's a huge amount. So anyhow, so erythrocytes are destroyed by macrophages in the spleen and bone marrow, releasing hemoglobin, which can be degraded to heme and globin. Now the globin protein can be catabolized into amino acids, and they can be recycled. Now, but in terms of, but there are challenges, because you have a huge amount of hemoglobin that is turned over a day. You have 6 grams, which is really huge. So there are two challenges. The first one is that porphyrin ring is hydrophobic. It's not soluble in blood, so it has to be dealt with delicately in order for it to be excreted or metabolized. Okay. The second thing is that iron must be conserved, and we already talked about that before.
And this is done through a number of reactions. The first reaction is very important. It's catalyzed by the enzyme heme oxygenase. We already talked about heme oxygenase in the previous lecture. Now, heme oxygenase metabolizes heme into a product known as biliverdin. Now, this reaction right here requires NADPH. That's 1. 2. Iron is released, and I mentioned that in the previous lecture. And then you have the production of carbon monoxide. This is the only reaction in the body where you have production of carbon monoxide. Now, biliverdin then goes through different reactions, and what's important is that the intermediates have different colors. That's why whenever there is a bruise in the skin, you can see that it goes through different colors. First, you have release of blood, hemolysis basically, and then heme is metabolized to different intermediates. Each one of them has a certain color, and that's why the bruise changes colors in here from blue to purple, green, and finally yellow. All of these colors are a result of the different intermediates of heme catabolism. Now, what happens is that biliverdin is converted to bilirubin, and bilirubin is then released into the blood, and it is carried by albumin. It binds to albumin, and albumin takes it all the way to the liver. Now, something that you need to pay attention to is that salicylates, salicylic acid, aspirin, as well as sulfonamides, certain drugs can displace bilirubin from albumin. So bilirubin becomes free of albumin and it can enter the central nervous system. Now the thing is, in adults it may not matter much, but in infants it can cause neural damage. So you need to pay attention to that. Now in the liver, what happens is that bilirubin is conjugated to glucuronides. So you have the formation of bilirubin diglucuronide. So this molecule is quite hydrophilic, and then it's transported into bile. Now deficiencies in either conjugating bilirubin with diglucuronides or transporting the bilirubin diglucuronide to bile can cause certain conditions, specifically Crigler-Najjar 1 and 2, as well as Gilbert syndrome and Dubin-Johnson syndrome. So what happens, as I said, you have release of bilirubin. Bilirubin is carried by albumin to liver. In the liver it gets conjugated, so you have conjugated bilirubin. Then it goes into the bile, where it can then be dumped into the large intestines. Now, in the large intestines, the glucuronide moieties are removed, producing bilirubin again. Now, this bilirubin can then be converted into urobilinogen. So right here you have urobilinogen. Okay? Now, urobilinogen, some of it can leak out into the bloodstream. Some of it goes back into the large intestines, and some goes into the blood. Now, the majority of urobilinogen is converted to sterocobalamin by bacteria. Now, this sterocobalamin is a brown molecule which characterizes feces. Now, the urobilinogen that is removed into the blood is taken up by kidneys, and in kidneys it is converted to a final product known as urobilin. Urobilin has a yellow color that characterizes urine. So, knowing the level of bilirubin is very important because it tells us a lot of information about the health of individual, specifically the liver. And this can be done by a specific reaction known as Vandenberg reaction. Well, what's important about this reaction is that it can be it is done in both water and ethanol or methanol. Now, the thing is, if it is done in water, then it allows for the measurement of conjugated bilirubin, the hydrophilic bilirubin. And this is known as the direct bilirubin. And it constitutes only 4% of total bilirubin. Now, if it is done in ethanol or methanol, these two solvents would allow for bilirubin to be soluble, and it can be measured. So what you have in here is that you're measuring all bilirubin molecules in a sample in order to know what the unconjugated bilirubin, the concentration of the unconjugated bilirubin, then which is known as indirect bilirubin. Simply you follow this formula right here. Total bilirubin measured from the organic solvents minus direct bilirubin, which is measured from the water-based reaction. Since we're talking about catabolism of heme, it's important to discuss jaundice. Jaundice is basically a group of disorders that are related to deficiency in the catabolism or transport metabolism of bilirubin, and they result in yellowing of the skin in individuals. Now, jaundice can be divided into two classes: either unconjugated hyperbilirubinemia, so there's increase in the amount of unconjugated bilirubin in these individuals, or you can have conjugated hyperbilirubinemia, so it's a condition where you have high level of the conjugated bilirubin. Now, let's look at the different markers related to jaundice in both of these types of hyperbilirubinemia. So unconjugated hyperbilirubinemia can be caused as a result of excessive hemolysis. So you have a lot of lysing of red blood cells releasing a lot of heme. That results in a lot of heme being converted to bilirubin. So you have a lot of conjugation of bilirubin. So you have a lot of conjugated bilirubin in here, going from liver to the intestines, and you have conversion of bilirubin to urobilinogen and sterocobalamin. Now, what characterizes people with hemolytic jaundice is that their stool would be very dark as a result of overproduction of sterocobalamin. But since the glucuronosyl transferase enzyme is overwhelmed with bilirubin, some bilirubin would leak out of the liver into the blood. Okay? Now, so what you will have here with hemolytic jaundice is that there is increased bilirubin in serum and increased unconjugated bilirubin as well. But the amount of conjugated bilirubin would stay normal because little conjugated bilirubin would leak out. Okay? And all of it would go into the intestines. Well, how about urine? Urine would look darker, and the amount of urobilinogen and urobilin would be high in urine as well, since a lot of urobilinogen as well as bilirubin would be taken into kidneys. Well, you can also have what is known as hepatic jaundice. So there is deficiency in the liver itself. There is a problem in the liver itself. So what happens in these conditions is that you would have increased amount or level or concentration of bilirubin in blood because there's leakage. There would also be higher levels of both conjugated and unconjugated bilirubin because, again, you have cell damage and release of both of these forms of bilirubin into the blood. Since we're talking about hepatic jaundice and we have a problem in the liver, there would be increased liver enzymes as well in the in blood. Urine would look darker because of the release of all of these forms of bilirubin into the blood, which are taken to the kidneys. Now, stool would look either normal or lighter in color, since there would be reduced bilirubin and its conjugates or its forms reaching the intestines. Well, another form of hyperbilirubinemia is known as conjugated hyperbilirubinemia. Now, what happens here is that there is production of conjugated bilirubin, but it does not get into bile or the intestines. So this conjugated bilirubin would leak out of the liver. So there would be increased level of total bilirubin in serum, and there would be higher concentration of conjugated bilirubin since it's stuck in the liver. It has to go somewhere, so it gets leaked out of the cells. liver enzymes are normal, and urine would look darker, okay, because there would be a lot of, again, bilirubin and conjugated bilirubin taken to the kidneys, which cannot convert them to urobilinogen and urobilin, and that results in reduced amount of these two molecules in the urine, and stool would look clayish as a result of inability of the liver to transport conjugated bilirubin into the intestines, so there would be low concentration of serocobalin in the intestines in feces. Now, jaundice can also affect infants as well. And the thing is, the enzyme glucuronosyl transferase enzyme that conjugates bilirubin with glucuronides is not very active when the enzyme is not active at birth. Now, its activity increases with time. So within like 2 weeks, the enzyme or the liver would be able to efficiently conjugate bilirubin with glucuronides. Now, the thing is, the problem is with premature babies. Now, what happens here is that the activity of the enzyme is not really that high, and that results in overproduction of bilirubin that cannot be conjugated, and this bilirubin would leak out. It goes into the central nervous system and it can damage the brain. So now, usually in normal infants, there would have high concentration of bilirubin, but these high levels are not toxic. But in premature babies, the levels would be very high. They can be very toxic. So a solution is to expose infants to blue light. What blue light does is that it breaks up bilirubin into a more hydrophilic molecule that can be eliminated easily.
There are two conditions that I'd like to finish with, and these are Gilbert syndrome and Crigler-Najjar syndrome. And both of them are caused by the production of a defective glucuronosyl transferase 1A1 enzyme. Now, if the enzyme is somewhat defective, it would cause Gilbert syndrome with mild asymptomatic jaundice. However, if the enzyme is not functioning at all, then it causes a more severe condition that is known as Crigler-Najjar syndrome. Okay. Now, what is the treatment? In young age, the best treatment is phototherapy. However, it doesn't work afterwards because the body gets bigger and light would not penetrate the skin. However, for older age, of course, the best treatment is liver transplant. Allah ma'akum.